So hi everyone, I'm Jeremy, and today is my final presentation about my final project, which is about the T cell analysis for space travel. And here I will be mainly focused on single cell transcriptomic by profiling of the T cell subset. And in the first step, I try to run a T cell subset cluster by the PBMC dataset from the crew members in I4 mission. And we can notice a lot of significant change among the different cluster. But in my project, I will be mainly discuss and focus on the increase in cluster 0 and the decrease in cluster 9. And later, next I ran a bar plot, try to confirm the decrease and increase among all the cluster. Yeah, in the T cell. And next would be the part of I used the find type markers function, try to identify the top markers in each cluster, and based on the information I got from here, we can do a cluster annotation, and based on this method, I can identify that the cluster 0 and 9 can be annotated as exhausted CD8 positive cell and also T helper 17 cells. And here I can generate my hypothesis is that after space travel, there is an increase in exhausted CD8 positive T cell and a decrease in the T helper 17 cells. And this feature is very similar to the patient with chronic viral infections. And during the space flight, chronic stress and microgravity may cause the CD8 positive T cell functional exhaustion, but also nutritional condition and circadian disruption will lead to the reduce of mucosal immunity and the inflammation control as the T helper 17 decrease. And in next step, I try to dig into more in a gene expression level within the cluster 0. So I ran again, use the find marker gene by identify what's the top genes in the cluster 0 changes. So here I can generate 3 biggest differential genes, and which they can be interpreted as reducing the T cell mobility and genomic instability, and also signal of terminal exhaustion. I think these 3 main findings could support my hypothesis about the CD8 positive T cell exhaustion. And next part, I run the geo enrichment analysis from the dataset. In here, I can also generate, like in the pathway level, I can also generate three functional change, like sensory related gene suppressed, which indicating the fatigue status in the group member's body, and also protein DNA complex activated, which can be identifying the DNA damage repairs process, and also in the NK cell mediated immunity activated part, this could be interpreted into immune compensatory mechanisms. And in the next part, I try to investigate in the cellular interaction level between Treg and T helper 17 to find out is the Treg X the inhibition on the T helper 17. So here we can observe that the significant increase in FOXP3, and which is key markers for Treg cell population, and also the significant increase in the IL-2RA gene, which can be identifying as the competitive inhibition on the T helper 17 cell. So the last part is the cross-validation, which I use the SOMA dataset to, we can find several data points in the plasma proteomics, which can support my hypothesis The first one would be the increasing of the PD-CD5 in the in the plasma could identifying a CD8 positive T cell exhaustion. And the second one will be the increasing IL-10 in the plasma could also showing the Treg exit inhibition. So lastly is my conclusion. Spaceflight associated physiology appears to impair and suppress the immune function in group members. And a key observation is that the expansion of exhausted CD8 T cell with a reduction of T helper 17 cell populations. And also multiple lines of evidence, including differential gene expression and pathway enrichment analysis and altered cellular interaction, all support this hypothesis. And the last point is, however, the I4 dataset compromised only 4 individuals, and the probably additional dataset would be necessary to validate these preliminary findings. Okay, that's all I have. Do you have any questions?